Thank you very much for this kind introduction. The title of my presentation is Mitochondria Transplantation, Reality or Fiction? A very difficult question which I will do my best to answer through my talk. I have nothing to disclose. First of all, to give you a quick outline of what I would like to talk about today. Well, first, in the first part of my presentation, I would like to explain the relationship between myocardial ischemia and mitochondrial dysfunction. I will then talk about the development of autologous mitochondrial transplantation and its current applications. And in the second part of my presentation, I would like to introduce you to my main research project, which I have been working on during my 2-year research fellowship at Boston Children's Hospital, where I've been trying to prolong right ventricular failure by transplanting mitochondria. So first of all, to really understand how mitochondrial transplantation works, we have to look into the relationship between myocardial ischemia and mitochondrial transplantation. So we all know that mitochondria play a major role as energy producers in all our cells, especially the cardiomyocytes in our heart. We also know that the mitochondria, that the electron transport chain is taking place in the inner mitochondrial membrane. But the really important thing is that the mitochondria can only fulfill all those tasks if they are supplied with enough energy themselves. So oxidative phosphorylation, which is providing our muscles with a high amount of energy, only works if mitochondria are supplied with enough energy themselves. So if the ATP levels in the cardiomyocytes are low during regional or global ischemia, the problem is that the mitochondria, which normally pump out calcium from the inner membrane, cannot fulfill those tasks anymore. And so calcium accumulates inside the mitochondria and makes them dysfunctional. So instead of this very high energy producing oxidative phosphorylation, glycolysis takes over, which produces much less ATP. So this constant low ATP levels alternatively proceed to irreversible damaged myocardium. So does mitochondrial transplantation solve this problem? In order to answer this question, we have to look into the development of mitochondrial transplantation, which pretty much starts in the year 2009, when Dr. McCulley, a biologist at Boston Children's Hospital, was the first to perform an animal experiment in New Zealand rabbits, where he induced an ischemia reperfusion model. So what he did was inducing regional temporary ischemia by snaring the LED for about 30 minutes, and then transplanting autologous mitochondria, so mitochondria that are isolated from the same subject, from a non-ischemic muscle of the same subject, into this area that became ischemic during those 30 minutes. What you can see in the graph up there is that the left ventricle developed pressure shows a remarkable drop during this time of ischemia in the two groups that have been experienced regional ischemia. We see the sham control, which does not show this drop, and we see that after releasing the snare, we see that during the reperfusion time, when the mitochondria became injected, we see that the mitochondria group that experienced regional ischemia before actually increased in left ventricular developed pressure and almost reached the level of the sham control. So after this initial publication by Dr. McCulley at Boston Children's Hospital, they performed a large series of small and large animal projects. They tried to transplant those mitochondria either intracoronary or epicardially. And they found that the transplanted mitochondria integrate into the cardiomyocytes within 20 minutes. And also they found that in the cardiomyocytes, the transplanted mitochondria increase the ATP content. They furthermore investigated and the fact that the transplanted mitochondria were still viable and still existed 4 weeks post-transplantation in the transplanted muscle. So far, no adverse effects have been demonstrated with mitochondria transplantation, and they have already been used, mitochondria transplantation in humans, to be exact, in pediatric patients that have been supported by ECMO more than 72 hours and could successfully be weaned from ECMO after mitochondria transplantation. As you can see in the figure on the right-hand side, the isolation process is rather simple, actually. You see we have to take a small piece of skeletal muscle, which is not much bigger than a 1-cent coin, and this small piece of muscle can be taken from anywhere of the body, from any skeletal muscle. They have shown that it makes no difference from which skeletal muscle they take those mitochondria. The only thing that has to be done is it has to be a healthy muscle that they are taking those mitochondria from. So after taking that piece of muscle and dissolving it in respiratory buffer and homogenizing this muscle, it gets digested by a bacterial proteinase and then filtered in 3 different steps, so that the entire isolation process can be done in less than 30 minutes. To show you a few applications that the Boston group has already been published or is already working on, as I already explained, we were working on different types of regional and also global myocardial ischemia. So we try to either transplant those mitochondria before the actual ischemia or afterwards in the reperfusion time, as you already saw. Also, they have been working on a project to preserve organs, mainly the heart, until transplantation was possible in order to prolong this time until transplantation. They have also recently published an article about acute limb ischemia, and I've been also working on a project with acute kidney ischemia in Boston, where we basically occluded both renal arteries for an hour and then injected mitochondria in the renal artery directly, and then it showed very good results. Also, I've already shown that we have used it already in humans, so in pediatric patients that could be weaned from ECMO. So moving to the second part of my presentation, I would like to show you the right ventricle hypertrophy or right ventricle failure model that I have been working on, and that I have tried to prolong failure with mitochondria injection. So right ventricular hypertrophy or right ventricular failure starts with pulmonary artery hypertension. So this can be caused by many reasons, but this increase in pulmonary artery pressure furthermore leads to an increase of pulmonary artery vascular resistance and afterload. To compensate for this increase in afterload, the right ventricle hypertrophies at the beginning in this compensatory phase. The muscle cells increase in size, and they try to compensate by increasing contractility. But if the afterload and the increase of pulmonary artery hypertension persists during this time, the right ventricle cannot compensate this increase anymore at some point, and it moves into dilation and failure ultimately. So this transition between hypertrophy and failure can be explained by a capillary rarefication. So the increase in size of muscle cannot be compensated by an increase of capillary vascularization. Also, the coronary perfusion pressure drops, and as I already explained before, the mitochondria become dysfunctional due to a lack of ATP in the cells. So to show you the animal model that we have done, we performed pulmonary artery banding through a left thoracotomy in immature Yorkshire piglets, so piglets that have been about 2 to 4 weeks old and 9 to 12 kilograms. And we have had a sham control in which we only performed a left thoracotomy versus the BA banded animals. In the BA-banded animals, we performed another intervention 1 month after hypertrophy. Through a subxiphoidal injection, epicardially in the right ventricular free wall, we injected mitochondria, or the respiratory buffer that I've already been shown that is part of the isolation process. So we had a vehicle group and a mito group and a sham control group, which consisted of 6 animals each. So to show you again the timeline that we used, again at the baseline, we either BA-banded our animals or they underwent sham operation. We performed an echo before and after the banding. And we also performed invasive PV loop measurements in which we measured the right ventricular outflow pressure, the PA pressure before and after the band. And then we followed those animals every 2 weeks. So the first echo after the banding was done 2 weeks after banding, and then the intervention was done a month, as I said, after the initial PA banding. And we performed another echo before and after the intervention. Then another echo was done 2 weeks after the intervention, and another echo was done at the time of euthanasia at the study endpoint, which was 2 months after the initial PA banding and 1 month after the intervention. And at this time point, we also performed invasive PV loop measurements. So we have a total of 7 time points in which we performed echo, sometimes epicardially, sometimes transthoracic echoes, where we measured different markers for right ventricular contractility. So we measured TAPSE and fractional area change. And we also measured the thickness of the RV free wall with MOs. In the invasive pressure PV loop measurements, we measured TB-TD max, which is also a marker for contractility, and we measured end-diastolic volume. After we euthanized our animals, we took biopsies from the right ventricular free wall, and we performed immunofluorescence staining, where we looked at hypertrophy and capillary density, and we looked at the mitochondrial shape in electron microscopy. So to show you just a few of our results, looking at the contractility of the 3 groups, you can see on the left-hand side, the TAPSE in yellow demonstrated the control, the sham control group. In red, the BA-banded vehicle-treated group, and in green, the BA-banded mito group. And you can see that at the baseline, they pretty much all started at the same levels. Then we see a drop in TAPSE and contractility 1 month after BA banding. And we have another increase in the mitochondria group, which almost comes up to the level of the control. Whereas the vehicle group has a significant drop in contractility at the study endpoint. A similar picture we saw in the fractional area change. Also here, the animals started around at the same level. And we have this drop in contractility after 1 month of banding, where at the study endpoint, the BA-banded mito group recovered and almost reached the level of the control. We also measured, as I mentioned, RV free wall thickness, and we found, not surprisingly, an increase after BA banding in the banded animals, which means that they hypertrophied. But then at the study endpoint, you can see that the BA-banded mito group further increased, so hypertrophied further, whereas the BA-banded vehicle group surprisingly had a drop in RV free wall thickness, which we explain in a dilation of the free wall. A similar result was demonstrated by the invasive PV loop measurements. We only have 2 time points, as I mentioned, in these measurements. We have a baseline and a time of euthanasia, and you can see that, again, at the baseline, all animals had around the same TB-TD max, and at the study endpoint, you see a significant drop in TB-TD max in contractility in the vehicle group compared to the control and the mito-treated animals. So looking at our immunofluorescence staining, I told you we took biopsies from the right ventricular free wall. Just looking at those pictures, you can see demonstrated in red desmin, a marker for muscle, and in blue we have the nuclei, and you can see that per amount of nuclei, the red increased in the BA-banded animals, vehicles and mitochondria. So we had a hypertrophy in terms of ratio per field of vision of muscle. If we look at the capillary density, we stained for endothelium marker CD31, which we can see is that in the sham controls and in the BA-banded mitochondria group, we see that the capillary density, the amount of capillaries is much higher than in the BA-banded vehicle group, which we explained why the BA-banded vehicle group alternatively dilated and went into failure. If we look at the mitochondria structure in the EM microscopy, we see that the sham control and the BA-banded animals show a nicely shape of mitochondria still. We see the crista, which are there in both groups. If we look at the BA-banded vehicle group, however, we see that mitochondria are swollen, the crista are reduced, and as I explained earlier, calcium accumulated inside the cell. So to summarize my results, we were successfully able to show that both our banded animals showed hypertrophy 1 month after we BA-banded them. And we could also demonstrate that the group that received vehicles proceeded further into right ventricular dilation and alternatively in right ventricular failure. However, by injecting mitochondria or by transplanting mitochondria, we were able to maintain the contractility of the right ventricle. We were able to maintain capillary density and to prevent furthermore dilation of the right ventricle and to prolong failure. Thank you very much.